So the last one, I didn't have the termination one. So the last step is going to be termination. Termination is where a stop codon is reached. So what happens there is that instead of a transfer RNA, a release factor comes in. Basically, it's kind of like an enzyme. You can see that the transfer RNA that is now in the P site, that was in the A site, still has the amino acids attached. But in this case, the release factor doesn't carry with it an amino acid. So what it does is it basically releases the last transfer RNA. What that does is it leaves the free polypeptide, which is like the native linear sequence of the amino acids. And the release factor also causes all the structure to come apart. In other words, the small ribosomal subunit, large one, and the messenger RNA is all released from each other. All right, so that's the termination. Now, just a couple of other things to talk about in regards to this. If we are looking at genes, let's say, for example, this is the messenger RNA piece up here. This was the original DNA, what they call the wild type or DNA. So if I have this messenger RNA piece, this would be my protein. So let's say, it's this short, all right? And then the stop codon would be the one that we reached that had the release factor, okay? So this is the normal protein. So there are things that can happen if there's mistakes in the copying of the RNA or DNA problems too, of course. So this one is what is called a silent mistake. So in this case, this A got changed. Instead of G, it's now A. But because of where it is in the codon, it didn't really change the amino acid. So this would be a silent change. There was no change in the protein. All right? So that's not really a problem. This one, missense, what happens here is there's a change, and that change will change the amino acid, in which case that protein will change. Now, in some cases, this is not bad. It could be good. That is, it could be, something that could be used by the organism. But in some cases, it just means that it's a protein that doesn't work, okay? And then there's the nonsense one where the change makes a stop codon immediately after the start one. So nothing happens there, all right? So that's of course a problem. Okay, so other things that can happen are, if we look at this, you can see, oops, got it backwards. Someday I'll get it correct. Okay, there we go. Okay, so this is the good one. Okay, so in this case, this is what we call a frame shift. Get off there. My cat is ruining stuff. Okay, so the frame shift is, what happens is there is an extra amino acid at the beginning of the code, all right? So if you have something at the beginning of the code, that can just mess up everything. It causes everything. So if that one wasn't there, it would be read AGG, AGG. But because of the extra one, it causes everything to be read off, okay? So in other words, it's going to be reading, that as a codon, and everything moved down. That's called a frame shift. In this case, it was just a nonsense frame shift. You could also have a frame shift that results in completely different amino acids all the way along. So the worst thing that can happen is a frame shift at the very beginning of a sequence. Okay, another thing that we want to talk about is the fact that as we make, you guys don't have to memorize all these parts, but this is just something to know. As we make the, as we make the protein, so here's the ribosome and it's making the protein. In this case, what is happening is it is feeding it into the endoplasmic reticulum space. And then when it gets into the space, it can then become the, do the folding and globbing, etc., and become a functional protein. Then it can be wrapped in the membrane here and sent out as a lysosome, right? So that would be like, to a food vacuole or something like that we've talked about before. All right, so some other things that we want to talk about. We want to talk about how do you do PCR? Okay, so PCR, or polymerase chain reaction, what that is basically I'm going to take a piece of DNA that I want. So let's say it's a gene that I particularly want. Maybe I'm doing a genetic test, or maybe I'm doing like some kind of test for like a crime scene or paternity or something like that. So I know kind of the area of DNA that I want. So I take that little piece of it, I cut it out, and then I'm going to put it in to a system where I can basically do replication. So I'm going to break apart the DNA strands. I'm going to go ahead and start copying the strands. They're going from either end there. That's called annealing. And then I can extend the nucleotides. Basically, I'm doing replication on both of these pieces. Okay, so that gives me more. And I do this over and over again. In other words, this is 2 cycles. I end up with 4. So I started with 1. I ended up with 2. Then the next cycle, I end up with 4. Then the next cycle, I end up with 8. So this is a chain reaction that is exponential. And it gives me lots of DNA that I can then run tests on. So I don't have to worry about, not having enough sample or something. So that's one method. Another thing to know about is gel electrophoresis. Now, normally we would have done this in lab and talked more about it in lab, but since we didn't have time to do it, we'll talk about it a little bit right now. So what happens in gel electrophoresis is that I have cut out perhaps a piece of DNA that I want to use for something like, again, maybe it's a crime scene or something. And I have gotten a little bit of sample there, and I collect samples from the suspects. And what I'm going to do is, run them through what we call a gel. So what happens in this case is I cut the DNA with what they call restriction enzymes. So restriction enzymes cuts the DNA into little teeny pieces. Some of them, though, are various sizes. So say, for example, let's just look at this coding here, right, because I have it written up here. Let's say I cut it every G. So in this case, this would be a really small piece, but this piece would be longer, right? So if I cut it every G, every guanine, then I'm going to have various sized pieces. And I'm going to put those into the gel. And what happens is, because DNA has phosphate groups on it, you might recall that phosphate groups are negatively charged. And they are going to then pull towards the positive electrode. So I'm going to load them on this end. This is the loading end, or the negatively charged end. And I'm going to put the DNA in here. And they are going to travel different distances based on how big they are. The biggest ones stay back, and the shorter ones travel further because they're lighter and they travel faster. Okay, so that's gel electrophoresis. In the lab and some of the genetics problems, you can kind of tell what we're doing. Make sure that you look at the lab ones, try to interpret them, and if you don't understand how to do it from the lab book, chat me, okay? All right, so that's that. Okay,